good morning, good afternoon, and good evening, Mr. Kenneth and Miss Ariska. Today I will be presenting about my review paper which I've made for the final exam for the Scientific Writing and Presentation Skills class. And my topic is referring to the industrial applications of recombinant Saccharomyces cerevisiae for proinsulin production. So, diabetes, it's a very well-known and very widespread medical disease, which is generally characterized by a lack of control over the blood glucose level within our human body. Now, diabetes itself actually has several causes, one of which is a high glucose diet, which causes the receptors within our cells to be insufficient or irresponsive towards high glucose levels, which leads to a lack of secretion of insulin, which causes the long-term effects of a high glucose level within the human body, causing it to rise uncontrollably. Now, diabetes itself also leads to other major diseases such as heart disease and kidney failure. Now, here's where the important molecule comes in. So, insulin. Insulin itself is an important polypeptide, which is generally secreted by the beta cells, which is located within the Langerhans inlets of our kidneys and, oh, sorry, pancreas. Now, the proinsulin itself is the one that creates insulin. So, proinsulin is a single stranded polypeptide chain, which serves as the precursor to the polypeptide, which is insulin. So, in a normal and healthy human body, proinsulin is converted from its native state into insulin, which is then secreted out of the human pancreas, which allows the blood glucose level to drop and regulates it. Now, how do we produce insulin? Well, a new challenger is Saccharomyces cerevisiae, which is considered to be the best or one of the best studied eukaryotic organisms that is often used in a wide variety of experiments, such as medical experiments, laboratory experiments, as well as other biotechnological experiments. Now, the reason why Saccharomyces cerevisiae is often used is because it could easily undergo recombination procedures, which essentially allows us to give Saccharomyces cerevisiae specific characteristics such as acting as a biosensor in a packaging, or as is in my review paper, as a method of producing proinsulin. Now, the main objective of my research or my review paper is to identify the industrial application of recombinant Saccharomyces cerevisiae in a wide variety of fields, such as medical fields and biotechnological fields, particularly for insulin production, as well as its effectiveness in producing strains of recombinant Saccharomyces cerevisiae that is capable of producing proinsulin proteins. Now, for the methodologies that I used three major databases from my research, which is Google Scholar, ScienceDirect, and Elsevier. However, aside from these three main databases, I also use some slightly smaller databases such as PubChem as well as ResearchGate. Now, for the query that I used, as you can see, I use several important keywords in order to locate specific literature, such as the keywords being used being Saccharomyces cerevisiae, as well as its more common name of brewer's yeast, recombinant or recombination, proinsulin production, as well as the industrial applications. So the methodology, of course, needs several criteria that must be met in order to ensure that the literature that is included is extremely reliable and consistent. So for the eligibility criteria itself, the first criteria is that the abstract of the literature must be available. Now, the reason why this is one of the eligibility criteria is that the abstract is essentially the overall summary of the entire paper itself. So by having the abstract available, it allows easier and more consistent narrative. Now, next, the Second criteria is that the research itself must also be written in English. The reason why I chose this is so that the literature that I use has a consistent flow. Now, there are also some additional criterions which I use within this research, which is that the research must include or cover the topic related to Saccharomyces cerevisiae, proinsulin production, as well as other industrial applications. Now, there's also one final criterion that I use for this review paper, which is that the literature itself must be from the year 2016 up until 2026, providing a 10-year total timeframe for the literature. Now, from the total amount of research that was found from the query in the previous slide, which is 325 research, a total of only 11 research were actually chosen from the query. Now, moving on to the discussion. So, for the first section of the discussion, it's from a literature by Kelton et al. From 2024, which talks about the molecular engineering of insulin for recombination expression in yeast or Saccharomyces cerevisiae. So, what they found was that insulin production increases significantly post introduction of recombinated microorganisms such as Saccharomyces cerevisiae. Now, why did insulin production increase dramatically? Well, it's because that before recombinant microorganisms were introduced into the market, people were essentially trying to produce insulin via big pancreases, which is less effective and less efficient in producing large amounts of insulin compared to recombinant organisms. Now, Saccharomyces cerevisiae itself also yields a very large amounts of insulin, as Kelton found that the annual yield of insulin by Saccharomyces cerevisiae recombinants is around 20 tons, which is supported by other sources, which states that the average is around 16 to 20 tons. Now, from the research by Kelton, there's also several factors to be noted, particularly about the insulin production in Saccharomyces cerevisiae. And those factors are the structural properties of insulin and proinsulin, as well as the biochemical and physical properties of proinsulin and insulin. Now, the reason why these factors need to be taken into consideration is because they heavily impact the secretion yield or the secretion amounts of insulin that is produced by Saccharomyces cerevisiae. Because when the team itself tested human proinsulin genes directly on recombinant Saccharomyces cerevisiae, they found that the yield or the secretion itself is actually quite low compared to other slightly modified insulin. So the main takeaway that we could take from this first discussion section is that the affinity for recombinant Saccharomyces cerevisiae in producing insulin and proinsulin is actually very heavily impacted by the proinsulin gene itself. And additionally, the changes to the structure also have been impacted. Now, next for the second part of the discussion, we also have the second research, which is by Wang in 2023, and he talks about the utilization of recombinant Saccharomyces cerevisiae in for diabetes medication. Now, what he found was that, first of all, recombinant microorganisms in general have a wide variety of usage. They're used in so many fields, such as medical fields, biotechnology, as well as other small industrial fields. Now, Saccharomyces cerevisiae in particular was highlighted by Wang, particularly because it has a high prospect due to its high tolerance towards unfavorable or harsh fermentation procedures. Some other organisms might not be able to withstand the harsh environments that is provided by the industry, thus making Saccharomyces cerevisiae a very highly anticipated type of organism. Now, additionally, Wang also noted how Saccharomyces cerevisiae has a low operational cost while comparing it to its high output of insulin. And lastly, Saccharomyces cerevisiae is also easy to undergo genetic modifications, which allows it to secrete specific recombinant proteins, in this case being insulin, of course. Now, Wang's research is actually contradicted by some other research which comes before 2023, particularly those research touches on the topic that despite Saccharomyces cerevisiae's affinity for producing insulin and its ability to be recombinated, it's actually still quite low in production. And comparing that to the high market cost of insulin, and well, it also has a high upfront cost with a limited scalability. However, this research did come before Wang et al. In 2023, and that makes a big impact because it indicates that there's already been a massive type of improvement in the usage of Saccharomyces cerevisiae in the industrial fields. So for the recommendations that I could provide, it's actually very simple. Future research could generally use my review paper potentially as a general guide to understanding the usage of Saccharomyces cerevisiae in a wide variety of fields, such as medical fields, biotechnological fields, and other industry fields. Additionally, insulin is essentially the primary usage for a lot of medication, and that Saccharomyces cerevisiae usage worldwide is actually extremely large. Now, for the conclusions that we could take from this review paper, it's actually quite simple as well. So, Saccharomyces cerevisiae essentially possesses the ability to easily undergo recombination procedures, which allows it to have a high affinity to producing specific proteins or molecules such as proinsulin, and which could be then converted into insulin for human usage. Additionally, Saccharomyces cerevisiae also remains to be one of the current best forms of producing recombinant proinsulin due to a high growth rate and high protein yields compared to other microorganisms. Now, additionally, Saccharomyces cerevisiae also contains and maintains the low operational cost with an ease in understanding the genetic recombination procedures to be specialized in making insulin. Now, lastly, one thing to note is that approximately half of the current worldwide insulin supply is actually still being provided by Saccharomyces cerevisiae, with more potential for it to be improved. That is all from my presentation. Thank you. And for the references, it is contained within the QR code. And for the AI declaration, I have simply used it for reference searching. Thank you and have a nice day.